connecting these dots is a part of omics technology. Also, omics is responsible for analyzing this data. So whatever we have collected, let's say just randomly, that earlier days when diabetes was diagnosed or was a new thing, that time people, how did they come to this was after assimilating data that fasting blood sugar increases, postprandial increases, HbA1c increases over the period of 3 months, and that data after connecting the dots, you label that as a disease called diabetes. So this is probably several years ago. Now the data has advanced, but it works on the same model and the same understanding that connecting each and every data that we have and make sense of that data is being done by omics technology. Now, what is omics? It is probing and analyzing large data of a biological system, and the whole technology revolves around central dogma of molecular biology. So the central dogma of molecular biology states that genetic data or information flows in one direction. That is, DNA will transcribe into RNA, RNA will make proteins, proteins will form metabolites, and metabolites will form biomarkers, and then there will be a function. So what happened before the 1990s? A human genome project was completed in the year 2003, but it was started in 1990s. So we had the first automated DNA sequencer in 1986 and protein sequencer in 1982-1983. The scientist who discovered these machines was Leroy Hood. And the information regarding whatever we collected looks like that before 1990s. And omics is responsible for the information to look like this. Although it is complicated, but at least it is structured. So, as I said before, the central dogma of molecular biology, DNA is responsible for making RNA, certain part of nucleotide chains that will give rise to RNA. RNA will code into proteins in the form of hormones, cytokines, antibodies, enzymes and transporters. And proteins will then give rise to metabolites like sugars, amino acids, lipids, organic acids, nucleotides. And then this comes into function or into a phenotypic form. So this is again, the reason why I am stressing on this is because after we understand each omic, we can elaborate on how it has implications in kidney health and disease. So, genomics is basically what may happen. Genomics is the study of genes. Transcriptomics is the study of mRNAs or RNAs, and it is what seems to be happening. So, DNA will make RNA, and not every region of DNA will be coded. So, the coded region will form a particular mRNA. So that is what it seems to be happening. Now proteomics is study of proteins. So what makes it happen? What makes a Krebs cycle happen, or what makes your thyroid function happen is the proteins. Now functional assays, that is high throughput assays, they reflect the biologic function. So that reflects what is happening. And metabolomics, which again we were discussing earlier, that is a part of metabolomics, and the study of metabolites is what actually happened. So what we see today is serum creatinine, serum GGT, SGOT, SGPT. These are all metabolites, and the study of that is called metabolomics. And currently, our focus goes reverse. Like we are more averse with metabolomics, and we are averse with genomics, but the data of genomics hasn't made sense yet. Every gene, we know that it must be responsible for some or the other function, but what is the importance of the gene? How can we utilize it? That is still in the experimental stage. So now there are some tools regarding omics technology. So genomics, it can be studied with DNA microarray. Standard sequencing is the first generation sequencing. next gen sequencing, and third generation sequencing. Then transcriptomics, basically RNA is studied through RNA microarrays and RNA sequencing. Proteomics, on the other hand, we make the use of mass spectrometry to study proteomes. And metabolomics, we do tests like liquid chromatography pairing with proteomes like proteomics like mass spectrometry to evaluate certain tests. Now, there is a drug which is commonly used in transplantation, that is tacrolimus. So the serum levels of tacrolimus are being done by liquid chromatography mass spectrometry, that is LCMS. So this facet of omics, that is metabolomics, is well engineered at present. We are also doing well at genomics, but what we routinely see as doctors is metabolomics. We are also seeing genomics come into play when we are doing HLA typing. So how we do HLA typing is by using SSOs. The primers, oligonucleotide primers are used, which tells us, establishes relationship between the donor and the recipient by doing HLA typing. So that all these tests which you might have heard of are a part of omics technology. Now, as I mentioned, these are the instruments. They are nothing like if you see, you feel that omics must be like a big room full of large machines, but it's not like that. These machines are automated and they're very compact, and a lot of centers in India have these machines. Majority of them are in the metropolitan cities, but I think even Aurangabad has a flow cytometer and a mass spectrometer, if I'm not wrong. So now let's coming to area of my interest, that is kidneys and omics. So as I mentioned earlier, omics begins with DNA. Now there are 46 chromosomes and there are 3 billion base pairs of nucleotides. So just figuratively, just to give you an idea, India's population is about 1.4 billion people, and we have 28 states. Similarly, if we take human genome, it has double the number of DNA base pairs. So imagine if it is a Herculean task to map down or jot down or track down every individual in the country, that is 1.4 billion people. You find out their occupation, you find out the way they behave, you find out everything they do. If that comes into perspective of omics, then DNA is being, the whole entire human genome project consisted of mapping down these 3 billion nucleotide base pairs. So basically, everybody is aware that DNA is made up of 4 nucleotides: ATGC, adenine, thymine, cytosine, and guanine. And this genetic material is what may happen, as I discussed earlier. It is not, suppose you have that gene, it does not mean that you may have that gene-related disease. But environmental factors will play a role for that gene to come into action. So DNA makes RNA. So the difference between DNA and RNA is adenine remains to be adenine in RNA. RNA is a single nucleotide chain. Thymine is, however, replaced by uracil, and then guanine and cytosine remain to be same. So when any transcription happens, a single DNA strand will transcribe into a particular code, like let's say ATGC, GGC, ATT, and then that will be converted into an RNA. There are several types of RNAs, but messenger RNA is the area of interest for transcriptomics. Now, the applied of genomics in kidneys is that because we have a lot of data has been studied on monogenic kidney disorders. That is, you have CAPU, where it is congenital abnormalities of kidney and ureteric tract. Then there is SRNS, that is steroid resistant nephrotic syndrome. Chronic glomerulopathies are also having monogenic roots. Even cystic kidney diseases like adult autosomal dominant polycystic kidney disease, nephrolithiasis, nephrocalcinosis, renal tubulopathies, even CKD of unknown origin seems to have taken roots from monogenic disorders. Now, the most revolutionizing gene that we have found through genomics is APOL1 gene, and it is said that if APOL1 gene, if it is present, the odds ratio, the likelihood of someone having, let's say, interferon-associated focal segmental glomerular sclerosis, the chances are that if you have that APOL1 variant, you might have nephrotic syndrome, or that is how focal segmental glomerular sclerosis manifests. In HIV-associated nephropathy in Africa, if you have APOL1 gene, the risk of having that disease becomes 89%. Similarly, the odds ratio goes on decreasing from FSGS, hypertensive kidney failure, lupus with collapsing features, non-diabetic CKD, transplant graft survival, lupus with kidney failure, non-diabetic CKD progression, and non-diabetic CKD in older adults. If you have an APOL1 variant, there are chances that you are at a higher risk of developing kidney disease. So there is a concept of genome-wide association studies. Similarly, there is TWAS, that is transcriptome-wide association studies, and then there is TWAS, that is proteome-wide association studies. Now, each study, I won't go into the details, but these studies, GWAS mainly revolves around SNPs. So SNPs is a single nucleotide polymorphism. What happens is, in a single DNA nucleotide strand, if one nucleotide is replaced by another, the whole sequence changes. So let's say we have an individual in pink, blue, and orange. So there is a SNP change, that is the A is replaced by G, G is replaced by T, and these individuals, and one more individual must be having another sequence. Now, SNPs usually occur, it is, thank God that they occur in the non-coding regions, 85% of them, but those occurring in the coding regions are responsible for diseases. So the most widely accepted, widely studied SNP is for diabetic kidney disease. So in that, if you have, but having a SNP in your body or being detected with a SNP in your body does not mean you will have that disease. It just correlates with that population. So what data says is that you can see on the left hand side there is individuals with disease, and on the right hand side individuals without disease. Now there is SNP1, those green dots that you can see, and then there is one without the disease. So having a SNP does not mean that you have that disease. But on the other hand, individuals with disease and SNP3, that is single nucleotide polymorphism 3, you can see individuals with that disease seem to have the population having that SNP is more common than the population not having that SNP. So we are still in the experimental stages. I am not the right authority to give you more facts about this, but this is in the streamline to make it available for clinicians. And if the patient asks, say, let's say somebody has adult autosomal dominant polycystic kidney disease, the prevalence of that disease is 1:400. So let's say a child is detected to have ADPKD at the age of 45 or 50. So if that the parent will ask कि अभी इसकी शादी तो करवा दी हमने, इनके बच्चों का क्या? So the highest test that is done for ADPKD right now is genetic testing. So if you have that polycystin 1 gene, polycystin 2 gene, the doctors can tell you the percentage, the possibility of you having ADPKD. It does not mean that you will have it, but the possibility increases if you have that expression of gene in you. Now, coming to transcriptomics. So, transcriptome, what it basically helps us is Currently, mRNA microarrays are being used in grafts, so they can diagnose injuries earlier. Similarly, for snake bite. But because snake bite data is more of from India and there is research still going on about that, but the most widely studied transcriptome injuries are lifestyle diseases, that is hypertension and diabetes. So what happens, what a transcriptome does is there is expression of antibodies against WT1. WT1 is a gene that is present in podocytes. And there is pan-CK, it is present in distal tubules. And CD, it is just present in distal tubules and not in proximal tubules of the nephron. And then there is CD45, which is the marker of immune cells. Now, in a transcriptome, spatial transcriptome profile of normal and diabetic kidneys, what they did is they took a sample of this kidney, they put the tissue in the wells, just like ELISA how we have, and then they attached a fluorescent antibody to it. The area that lighted up showed that podocytes are present, pan-CK is present, CD45 if it is present or not. So this the colors are not appreciated well, but for instance, this is a small tubule, it is marked by yellow. So yellow, the color yellow, that means pan-CK is present on those distal tubules. So transcriptome is basically to identify these molecules if they are present in that differential part of the nephron. Similarly, how diabetic kidney disease affects various structures in nephron. So this is a normal healthy glomerulus. As you can see, there is proximal tubule, distal tubule. So the relevant proteins or the relevant mRNAs that are found in these structures, they may not be found in unhealthy individuals or that individuals suffering from that disease. So transcriptome will help us label that. This is just not for biopsy but also for blood and urine samples. Now, as I mentioned earlier, another slide where there is kidney specific data from full transcriptome atlas. We have a human transcriptome atlas and we have just taken kidney specific data. So you can see that this is a healthy glomerulus and these are glomes here, but there is no uptake of that antibody or antibody is not attached in those glomes, so this portion is abnormal. So this was done in the rats, not in humans. So you can see there is abnormal glomeruli with having very fewer podocytes and more immune cell infiltration that you can see. Whereas on this side, the normal glomerulus, the cells are not these pink and yellow cells that you can see. These are representative of immune cells and pan-CK cells that are there in the distribution. Now coming to the next omics, that is proteomics. So we have four diseases that we commonly come across as nephrologists, that is membranous nephropathy, FSGS, amyloidosis, fibrillary glomerulonephritis. These are all histopathological findings. They are not the disease per se. They are present as proteinuria. Now, if back then, like 10 years back, if you had proteinuria and you had a electron microscopy, only then you would be able to differentiate between membranous and FSGS or even nephrotic syndrome for that matter. But now, by just doing serum plateau levels or serum TH7, thrombospondin type 1 domain 7A levels, you can just say that this patient has membranous. In fact, plateau level serology correlates very well if you are having membranous nephropathy. You can skip kidney biopsy. So that is one. Then FSGS, there is a protein called SUPAR, soluble urokinase protein, that it is still in the experimental phase. If those levels are raised, the person is supposed to have FSGS. But of course, biopsy will be more diagnostic. Will help us diagnose the condition better. Similarly, then there is this condition called fibrillary glomerulonephritis, the subset of amyloidosis. The protein there is DNA JB9. So if that protein is found in the biopsy, everything looks the same in amyloidosis. You have just a pink dot in the tubules or a pink dot in the glomerulus somewhere. It is an amorphous-like structure. And you don't know if it is an amyloid, if it is a subset of amyloid. You have to do electron microscopy to find out how it is arranged. And one electron microscopy costs around 9,000 rupees. And we have electron microscopes only in Delhi, Mumbai, and I think Parvati. Oh, okay. So that is a surprise. So electron microscopy is not for all. We have to send that sample, and the report comes after 45 days. And let's say, unfortunately, if you don't have an adequate sample, they send the biopsy back, and you don't have a answer to your question. So this is proteomics. These are all the genes, basically transporters that are present in various parts of the nephron. That is SLC16A1. These are transporters, and there is a particular gene responsible for it. But this data still has to make sense in the clinical world. Now there is a wide study on urinary proteome. It's not just done for kidney diseases, but even for cancer. So it is said that, or the study says that urinary proteome reflects 70% of the urogenital tract. Suppose you have a urogenital disease and you find a molecule in the urine, there is a 70% likelihood that that molecule will be found in the urine because it comes from the kidney or urinary tract. But if you take the same urine for another disease, 30% of that molecule that is circulating will come to the urine. So the likelihood of detecting someone of a disease through urinary proteome is 70% for urogenital tract diseases. But it is a very good number, is 30%, where let's say you have a cancer cell, the blood passes through kidneys, whatever is in the circulation will go into the urine, and that 30% can be reflected in the urinary protein. So that is about proteomics. Now, renal metabolism. In biochemistry, we all study about Krebs cycle, we all study about urea cycle. Then I never found it interesting. I couldn't mark it up at all, but we had to write it in our answers. So, but I also did understand that how it is important. But in fact, whatever cycles we have learned in biochemistry so far, they've all been tracked, and each metabolite of that cycle will reflect, or the levels reflect, a certain entity amongst these. So let's say you have AKI. In AKI, the beta oxidation pathway, there will be acyl carnitines that will be increased. So like Sir said about sleep file, in NGAL, basically it reflects injury due to ischemia, injury due to sepsis, injury due to drugs, and maybe venomous. But NGAL cannot be elevated, or actually NGAL cannot be used as routinely as we use serum creatinine, because we still don't know in what all conditions it may come false positive. So the metabolomes are responsible to give rise to biomarkers. So in AKI, most commonly beta oxidation pathways such as acyl carnitines. When there is oxidative stress, homocysteine levels rise. If you have hypertensive patient, homocysteine levels are down. Or you have a stroke patient, they do have homocysteine levels, they are elevated. Then in CKD, there will be indoxyl sulfate that is elevated, ADMA, SDMA. Then end-stage renal disease, that is when irreversible damage has occurred. Again, these molecules are increased. So every disease will have a specific molecule that is increasing. But it is out of our scope of practice to do this, because if we do this, it will probably cost somebody his entire life savings or even more to do these investigations. But they are in the experimental stage, and they may come in the clinical side where we will be able to diagnose better. Now, in kidney transplantation, lactates are elevated. Even in critical care cases, lactates are elevated. So, we are using certain molecules to see how day-to-day progress of that injury is unfolding. Now, renal metabolomics. This is one just example figuratively I thought would be very interesting. What they did was they did all these tests on T0 of transplant day. That is just before doing transplantation, they did these levels. And of course, the kidney before undergoing transplant has undergone the brunt of long-term hemodialysis process, or it has had several influences of other body mechanisms that happen to that organ, and even ischemia reperfusion processes that accompany the metabolic changes. So what they found was that glucose valine alanine, they are reduced at T0 time. T1 it will be different, T5 it will be different, T10 it will be different. And mannitol, purate, adenine, creatinine, lactate, citrate, histidine, all will be elevated before transplantation. And after transplantation, when you do these levels, there will be a drastic paradigm shift that indicates that probably this kidney is healthy and this kidney has taken over the functions of that individual body. Now, renal biomarkers. This is the last topic after metabolomics. So, renal biomarkers, as Sir has already mentioned, the way we have serum creatinine, which is not affected, which is secreted, which no other organ is making, and it is only exclusive to that organ. So, biomarkers have that kind of a speciality of characteristics in them. All these that you can see, they all have a characteristic attached to them. So, NGAL, like I said, is responsible for ischemic and septic cause of AKI. But they may not be elevated in any other illness. Similarly, now you can see NGAL and CKD also. So, NGAL in AKI and NGAL in CKD, you cannot differentiate, but they are elevated. Similarly, there is kidney injury molecule 1, KIM1 in AKI and AK. In CKD, nephrotoxicity also, like drug induced also, KIM1 will be elevated. So you don't know what pathology, let's say even if somebody has snake bite, but that snake bite also has diabetes. So you never know if that NGAL is due to diabetic kidney disease or it is because of the AKI. Then we have liver-derived fatty acid binding protein. Again, there is an overlap, and then there are microRNAs. So urinary microRNAs are extensively being studied, and they in higher setups like wherever these molecules are being investigated, mostly in the United States. MicroRNA, they are using graft tissue and they are attaching microRNA to find if there is graft injury. Protocol biopsies are done everywhere. Not necessary that individual will have any illness or they do protocol biopsies at a particular interval. Let's say day 0 they do a biopsy, after 3 months they do a biopsy, after 1 year, after 5 years. So when they serially monitor, they can see microRNAs of that data. And all this data is stored. So you can go back and pull out whatever information you want. Now last, advent of omics technology that is still, it's a wonder that is going to happen in the coming years. That is body on chip. So, in fact, when I was in Hyderabad, I also witnessed one experiment where there were 2 pigs that were being studied. One pig was the patient suffering with kidney disease. He was hydrogenically embolized. His blood vessels were embolized, and kidney disease was induced in that pig. And there was another pig who did not have any illness. That pig was called Buddy. So, it was called allo-HD. Allo-HD is basically when a recipient's blood is exchanged by the buddy's blood. So, we don't need expensive dialysis machines. Only 2 humans, instead of undergoing transplant, if you connect with the phlebotom, like the way we do blood transfusion, we connect with their vessels. The person who has kidney disease, his or her blood will be purified by the person who doesn't have kidney. It is still in the experimental stages, but This is where the future is right now. And so body on chip is basically what they have done is every organ is on a chip. So this chip is made out of a polymer, 3D polymer, and there are live cells on it. If you ever get to see the video of it, it's very interesting. These channels which you can see act as lumen. So this is like blood counterpart, and this is air or even urine or any metabolites that can be excreted in the tubules. Now, this chip is connected to a computer, and that computer can simulate the entire pathophysiological process on this chip. Now, why this organ on chip has come into place is because animal studies, a lot of PETA activists don't want animals to undergo all these painful studies on them as experiment guinea pigs. So what they did was, after you make this chip, you can understand how a cell will behave, how the cell will, its physiology, pathology, even how it will act after a drug is given. So you no longer need to have those phase 1, phase 2, phase 3 clinical trials, the entire process. Instead, after this becomes successful, you can see the drug is administered and there are live cells on this. Whatever interest we want, they will show the reaction, or we will know how they will behave. So that is organ on chip or body on chip. So we have blood barrier, brain barrier on chip right now, lung on chip, gut on chip, heart on chip, kidney on chip, liver on chip, and body on chip. So the video I saw, for my understanding, was lung on chip. What they did was, it is just mind blowing, is that they line tissues, the lung cells on this chip, and you can actually see that chip is breathing. The moment oxygen air is given, you can see that the chip inflates and deflates. So that kind of a study is already in process. So that is body on chip. So with that, I think I'd just like to summarize how omics has made life of a clinician easy and complex at the same time. Because even if we have all this, but if we don't know how to utilize it, so if anybody who has the privilege to get diagnosed also, even if that person is not satisfied, I have kidney disease, dialysis he answered it, transplant he answered it, but I want to know more. If they have the privilege, they do all this. But we have not reached any stage where we can modify that even after knowing it. So that is still under process. So in summary, genomics is study of genes, transcriptomics, mRNA, proteomics, proteins, metabolomics is the metabolites that we discussed. These give rise to biomarkers. Even proteomics gives rise to biomarkers. And then we have a phenotype. Thank you.
Thank you for our very nice and detailed lecture. You have shown facilities.
Very excellent talk, super special.
Thank you.
It is more than that.
I have one question. The presence of omics does not mean that somebody has a disease. It's only after some environmental hits.
Yes.
I was interested in knowing what are the common environmental hits, and should we employ this omics in all patients of such hits?
I think next topic is.
The answer to that question is, suppose there are two severely obese people, for example. So with this technology, you know whether the adult is in front of you is genetically modified versus he was genetically normal, but he has eaten too much. So knowing that you have syndrome X and gene modified acid-related genes and the metabolic syndrome, the CDP of central obesity versus your genes were normal, your mother when you were in the womb, your epigenetic changes and intrauterine programming was not done, but you had your lifestyle made you push into obesity. So that's how this the adult who has genetically programmed modified obesity can be picked up by this technique. So each disease, in kidney diseases, majority of the disease FSGS, nephrotic or CKD, you will have a genetically beginning disease and some epigenetic factor or some environment factor that has hit him, and then he has presented to you as CKD. Versus the genes were normal and something else has hit him and he is presented again. So with knowing these two different group of people will be very important for treatment. Treatment will be totally different.
Just to add on, Angelina Jolie had BRCA1 or BRCA2 positive, and her mother had breast cancer. She didn't develop signs of breast cancer. She didn't even have any symptoms also. But just because she had the privilege to undergo genetic studies, she underwent that and she was positive, and she had a mastectomy without having any symptoms. And I don't know if that is foolishness, but being so empowered or being so woke that you take steps so that वो होना ही नहीं चाहिए. That is the level of understanding or awareness that is there in certain parts of the world, and we have to still reach there.
And we also actually feel it.
Yeah.
And it's also actually, yeah. Where do you place this in?
In proteomics or metabolic, mostly in proteomics, because it is antibody is in protein.
The antibody is a new branch of proteomics called antibiotics.
And there are so many omics now that this is like just a one-minute problem. Cities and villages still have to come for each omic.
Economics मध्ये नाही आहे.
Epigenetics आहे, epigenomics आहे. Everything has a subset. And
How the genes are going to express in an individual, that is also to be explored. What Angelina Jolie did, that cannot be applied generally.
yeah.
This or in any branch. That is the thing.
That is something that God's keeping his hand.
So, like science has plus points also and negative points also. So one need to understand all these things. How many movies are applicable in city like all?
We are doing regularly for transplant. We are exploiting omics like anything. In fact, a whole transplant workup that is done is on omics. We have HLA genotyping, we have lymphocyte cross-match, we have even drug levels that are being used by these tests.
And last half an hour, it's like we were listening to polypersonal genes.